Meeting is streaming live on custom live streaming. देखो चाहिए तो तापर सेटा कोन क्लोज करियो पड़ियो परा कानू सर
हेलो सर
हाँ हाँ एबे लाइव ऑन लाइव स्ट्रीमिंग हल्ला बोले देखो जी हाँ से येटा केंद कटियो ए जो YouTube टा केंद कटियो सर सर स्पीकर जॉइन कर ले
Good evening, sir.
Sir, good evening, sir. Dr. Subrata, can you hear us?
Yeah, we can hear you. Yeah.
Dr. Subrat, this is Dr. Ravi Kumar, Principal, Roland Institute of Pharmacological.
Good morning, Dr. Kumar. Good evening, Dr. Kumar. How are you doing?
I'm fine. How about you?
Very good, very good. Thank you.
Thank you for accepting our invitation.
Thank you. Thank you for inviting and getting reconnected with Behrampur after a long time. I'll speak about it about a minute at the beginning when I start talking.
Definitely.
Thank you, Dr. Subrata, for accepting our invitation. Okay,
Thank you. Thank
nice
you, Dr.
feeling to see you after a long time.
Yes, absolutely. Absolutely.
So what is the time right now over there?
It's just 1 minute away from 8 o'clock in the morning. Monday morning we have here.
Sorry for troubling you.
Oh, no. No, that's my usual routine. I am an early person, always. Usually by this time, I start for the university. Usually by 7:50, 7:55, I start for the school. Today, I'll go after the talk, then I'll start driving.
Okay.
So that's the only difference. Otherwise, it's same for me. No change.
So is the school open for students?
No. It's virtual till August, but I go for first 4 months. I did not go because it was quite bad. We had a really, really bad COVID situation until January. We had daily 10,000, 15,000 cases in the state, 15,000 daily.
Oh, in the state itself.
Yeah, in the state only. And out of that, the region, South Florida, is like almost 75% of those cases. So we are heavily affected, but that's why I go for last 6 months, but students are off campus from last March, and faculty also only 70% or 30% comes on campus. So research labs are also somewhat affected. Only limited people do because we have a restriction of 5 people at a time in the lab. Only 5 people.
Shall we start?
So another 2-3 minutes after that we will start. So is there any live demonstration, Dr. Subrat?
Yes, at the end I will have the software to demonstrate That just gives some flavor in general.
Okay. Right.
So yeah, I'm going to use probably the last 30 minutes or so for that purpose.
So I request Mr. Kanucharan Panigrahi sir to introduce our speaker to the participants.
Good evening, respected dignitaries on this web platform. I am Mr. Kanucharan Paningay, Assistant Professor, Roland Institute of Pharmaceutical Science, here to introduce today's speaker, Professor Subhrab. It's a privilege for me to introduce Dr. Subhrab. He is working in College of Pharmacy, Lakin University, USA. Dr. Subhrab is an alumnus of College of Pharmaceutical Science, Mahuda, Barampur. Subsequently, he obtained his M.Pharm from Bombay College of Pharmacy, Mumbai University. Finally, Dr. Dev received his PhD from the University of British Columbia, Vancouver, with drug metabolism and cytochrome P450 enzyme as the main focus. He identified a novel cytochrome P enzyme named cytochrome 2S1 in his PhD thesis work. Subsequently, his translational research in Vancouver Prostate Center at Vancouver General Hospital highlighted the effect of drug metabolism and pharmacokinetics on prostate cancer, drug interaction, drug efficacy, and adverse effect. Currently, Dr. Dev is the chair and associate professor in the Department of Pharmaceutical Science, Larkin University, College of Pharmacy, Miami, Florida, USA. He has published more than 60 papers, 60 peer-reviewed publications, and has more than 75 invited talks and conference presentations to his credit. Dr. Dev served as a peer reviewer for multiple journals of reputed, including European Journal of Drug Metabolism and Pharmacokinetics, Biopharmaceutics and Drug Pharmaceuticals, and expert opinion on drug metabolism and toxicology, and also served on different journals as editorial board member. With this, I welcome you, sir, on this web platform to address the gathering. Thank you, sir.
Okay. Good evening, everyone, and thank you, Dr. Panigrahi, for giving this kind introduction. Let me first share my slide, and then we'll start talking. Okay, so slides are visible now, right?
Yes.
Okay, sure. Okay, so again, thank you to the organizing committee, and I would like to again extend my thank you to Dr. Kumar for inviting, and obviously my connection with Behrampur goes back with Dr. Niranjan Patro about 25 years back. And as you have just seen, that essentially I belong to northeastern part of India and Tripura, where I did my schooling, and then it was College of Pharmaceutical Sciences. Give me a second. Some reason the slides are not moving.
Yes,
Okay.
sir.
Yeah. So subsequently, I did my. I was in Bahrampur in '96 onwards for some time, obviously, to complete the undergrad in CPS Mohoda, and then that's the time my affiliation goes back with Dr. Patra, Niranjan Patra, and then Mumbai University, and subsequently we have already heard those things. But the key part, I think, over this pandemic, what happened, the pandemic is not good at all, we all know by now, but digitalization or technology, our ability to connect with people have increased during this time. And in spite of sitting about 9,000 miles away, I think I am anticipating a very good session. And if you and that's where I am here, and you are in this southeastern part of India right now. So I understand this is an AICT sponsored training program. So more than talking about research, my goal of this presentation is to provide some tools. And I understand majority of the attendees are either faculty or PhD fellow, right? That's what the attendees are, right, Dr. Patra?
Yeah, yes, yes.
Okay. Okay. So I thought this is a very good platform to share something which attendees can consider applying moving forward in their research and teaching and whatnot. And then subsequently, I'll be also available later if you need some help in terms of implementing any of these drug research or pharmaceutical research tools. So you are going to see a lot of concepts and application towards the end. Last 30 minutes, I'm going to save for showing you a software related to the topic today. About towards the first hour, end of first hour, I will also open for questions, so that before we get into the application part, showing you some of the examples, I can answer some of the queries or doubts, and then we'll finish with the application, and again extend a brief question towards the end. So with that, let me start with that. Okay. So today's conversation is about models, different experimental models of Cytochrome P450 based drug metabolism and drug interaction. So the main focus is of drug metabolism and how we can apply it in different areas, including drug interaction studies. So the objectives of this discussion today will be to understand the role. What is the role of drug metabolism in drug development or new molecule development, and as well as therapeutics? We were trained primarily to think about drug development, but as you know, in India and especially in US from long back, therapeutics is also a big area of research in the university level, as well as in the hospital level, and in the industry as well. So how drug metabolism is interpreted in different areas or applied. Recognized different experimental models, different types of tools are used to accomplish the outcomes from this type of metabolic reactions. So we'll see what are those. Then I'm going to run you through a different steps of a drug metabolism assay. How do you run a drug metabolism assay? What are the basic steps? Obviously, we won't be going through all the details here, but a snapshot how drug metabolism assays are carried out, and then finally the computational models. When we were in graduate school, means MPharm and PhD, computational models were mainly for drug designing, right? How do you design a new drug molecule and whatnot? But over the last 10 years, this particular area has expanded like anything. Metabolism and drug interactions and pharmacokinetics. So we will talk towards the end how you can use different software for predicting drug metabolism, pharmacokinetics, and drug-drug interactions. So that's the software we'll be focusing later, GastroPlus simulation software. Okay, so that's the outcome I am envisioning today by end of this session, and then subsequently whatever help is needed from the attendees. So in terms of sequence of pharmacological actions, and this is a self-explanatory, and I realize here most of the people are faculty and so on. So I will not get into the basics of it. I'll just give overview, and wherever it is new concept, I feel I'll spend more time. So when you take a drug, it goes into systemic circulation by absorption, right? And then some of those get distributed in different tissues. And then some of those, that fraction goes to site of action. But a key part is also lost in the elimination. And drug metabolism by metabolizing enzymes has a key role in deciding the drug concentration. How much drug will reach to site of action to cause the pharmacological effect, be it toxic or be it effective, therapeutic purpose. But end of the day, this is the key part. The drug concentration is affected by elimination from drug metabolism. So some of those enzymes can control how much drug will reach to, for example, brain, or how much drug is going to reach to the, for example, lung in some cases, whenever you have lung-specific drugs. For example, during the COVID, you see a lot of those. So overall, the key part, drug metabolism has a very important role. And why it is like that? So metabolism and biotransformation, you can consider in two components, phase 1 and phase 2 reaction. So xenobiotics, whenever we take a medication, or it could be a toxicant like pesticides, some of the dietary compounds, phase 1 enzymes add a polar group, oxidation, reduction, hydrolysis reactions, and that makes those lipophilic xenobiotics more hydrophilic, more water soluble, and that's how they get excreted from the body. If they are not getting through that addition of polar group, water soluble functional group, we are not going to see the excretion, and it's going to accumulate in the body and cause toxicity at some point. Phase 2 also has a similar purpose, because some molecules they need a conjugation reaction in the phase 2 step, so that it becomes more water soluble, and eventually they become more hydrophilic, and they can be easily excreted. So that's the whole purpose of metabolism, to make it more I'll say water soluble, for getting rid of from the body and avoid any toxicity subsequently. And I think I already mentioned some of the functional groups which can make things more water soluble, like hydroxy, amine, carboxylic acid. This is a very basic thing. We all know that. Conjugation usually are done with the amino acids, or things like glucuronic acid in the phase 2 step, that makes things more water soluble. They are more hydrophilic, and that gets out of the body. Now, in terms of phase 1 and phase 2 enzymes, phase 1 is the part we'll be focusing today, obviously, most of the time, but I will be also referring some phase 2. Cytochrome P450, or in short, CYPs. CYP is the acronym, that's how we call it. The ones which are rounded with the green oval, CYP3A4 or 5, 2D6, 2C9, 2C8, 2C19, these 4 are really major enzymes which cause phase 1 reactions. Whereas under phase 2, you see UGT is a big player, such as uridine 5-diphosphoglucuronatransferase, as I wrote here, UGTs. And the pie chart tells you that UGTs are about 1/3, at least 1/3 of the contribution of the phase 2 enzymes. Then you have sulfotransferases and acetyltransferases and so on. So different types of enzyme is not our discussion point today, but the models we'll be focusing on. But I wanted you to see what enzymes we are talking when it comes to drug metabolism. Same way, CYP3A4 is almost about 50%, as you can see. It has a contribution of 50% from the green pie of the left, you can see, and so on. In short, these enzymes on this slide are essentially taking care of about 97, 98% of the drugs in the clinic. Together, they excrete the drug. So that's why it is important to carry out that type of study as you develop a molecule, or you go for a clinical studies and so on. Now, a very brief snapshot of the cytochrome P450 or CYP enzymes. As I said, the acronym is, that's what we are using. So it's a superfamily of enzyme. It has more than 50, about 57 enzymes actually. And they are located in the endoplasmic reticulum. But in terms of tissue, the liver, small intestine are the major players. They take care of probably 90% of the metabolism. Kidney, adrenal, prostate, and so on, they are other CYP expressing enzymes. Now, in terms of substrates, all variety of molecules can be metabolized. Drugs, medications, antibiotics means vitamin D, vitamins, hormones, all of them. Environmental toxicants such as pesticides, environmental pollutants, all those, cigarette smoke, all of them can metabolize, be metabolized by these CYP enzymes. At the end of the day, their function is either to eliminate lipophilic compounds, we just talked about it, or in a very select case, actually, biosynthesis or elimination of steroids and vitamins from the body. So you do see that they have a very wide range of function, either metabolize drugs and eliminate, or some cases biosynthesis and then eliminate some of the steroids. But I'm not sure if you ever thought about it, because CYP enzymes are part of biopharmaceutics and then pharmacology in both cases. Their name is very strange. The nomenclature is very strangely done. CYP3A4. So and often it is not clear. I have seen especially with students. The way that it is named, CYP is the name of the superfamily, as I said. Now, the first numerical 3 is the name of the family they belong. Then the next letter tells you, the alphabet tells you the subfamily. And then finally, the last numerical number, in this case 4, tells you the isoform or protein, CYP3A4. For example, there are also family of 1, 2, 4, and so on. So that could be CYP2C1, means the family is 2. Now, the subfamily also can be A, B, C, depending on the family you talk. So, for example, 2C9, 2CEC is the subfamily. And then 9, for example, or it can be 1, 2, as you move along. So, in short, this nomenclature is done to highlight their family, subfamily, because based on their amino acid similarity, they are typically named in different. They are named in different families or subfamilies, and that's how they are characterized. So eventually, the basis of classification is amino acid or protein sequence similarity. Okay. Now, I mentioned about this earlier, but I wanted to show a pictorial diagram that drug metabolism is important in the drug discovery and development in different stages. For example, initially when you go for IND and whatnot, you see the ADME studies. Even before that, when you are doing the discovery phase, look at the bottom, rapid screening for CYP inhibition, induction, and so on. And then again, during the preclinical development, during the IND phase, you see CYP reactions. Once you go to the phase 2 and phase 3 clinical areas, you need to do clinical DDI study, which is again drug metabolism based, transporter based, and whatnot. But metabolism is the main source of drug-drug interaction, as you realize. So even from the lead selection part, you have application of these concepts. And with the software I'm going to show you towards the end, actually it is used pretty early on during the lead selection, because you don't want to waste your resources and figure out in phase 2 or during IND that there was a problem with the molecule. And CYP inhibition especially is a huge issue. A lot of drugs have been withdrawn from the market in the past when this area was not that developed because of inhibition of some, for example, cardiac enzymes. If a drug is inhibiting a cardiac enzyme or a drug is inhibiting some CYPs which are critical for a drug interaction, you need to be careful and you need to know that information right from the beginning. So in short, starting with the drug discovery all the way till post-marketing, you have the role of drug metabolism, either in the chemistry part at the beginning or at the therapeutics or DDI part at the end. So its length and breadth is quite wide. Now, in terms of purpose of the drug metabolism studies, some of them I mentioned, but I also wanted to list here, is primarily to identify the metabolites of different drugs, herbs, and some of them could be toxic also. And if you have a drug with metabolite as toxic, you need more investigation. Otherwise, the FDA is going to reject it. And later on, you have more liability when it goes to the market. So at the end of the day, it gives an assurance right from the beginning, the safety of profile of the drug or therapeutic success of the drug. Now, next one is the characterize the primary enzymes. If I can request the attendees, would you mind please put on mute so that everyone can have the step one streaming. Hello. Okay. Muted everyone else except me, so I think that should be fine. So thank you for doing that. So that way, everyone will get the same message coming through this discussion. To characterize the primary enzymes responsible, early on in the drug development, we want to know what are the enzymes which are responsible for its breakdown, because it tells us, will that drug be toxic down the line, or will it cause drug-drug interaction, right? And that's what the next point, toxic metabolite cellular conjugates. The other one, you have effect of induction or inhibition when you get exposed to drugs or dietary substances, how our body enzymes behave over our lifetime. We can understand by knowing the drug metabolic profile in different models. DDI, and so not only drug-drug, and this is the key part I want to highlight. We always think drug interaction, or most of the time we think drug interaction is always with drugs. Natural products, I have spent significant time in research regarding drug natural product interactions, because it is a general mindset that natural products are all safe. Although I am a big believer and supporter of natural products, but we can't ignore the fact that natural products can do, and it does do, a lot of drug interactions. So it's not only drug-drug, also drug-herb or drug natural product interactions are important to study through these tools. Polymorphic enzymes, when it goes to pharmacogenomics or genetic effect of enzymatic changes, you need to know, because that's why drugs which can work in a population of United States may not work in India or in some African countries. So I think the key part is that polymorphic enzymes are important to know and their role, so that you can understand the inter-individual variation, how they differ between individuals, one person to another person, and accordingly you can take steps. You can give recommendation to the individuals. Now, in terms of experimental model, in terms of experimental models, there are 4 types of things, and now we are entering into the key part of, or the main part of the presentation. There are 4 different segments of experimental models that metabolism can be studied. First one is in silico. That is usually the first thing to do. But since I have some application to show, I will save it for the end. I'm not going to discuss at the beginning, but typically when you start a project, you start with the in silico, because that is the most cost effective, because it's computer based and you don't need a lot of manpower. You don't need a lot of money to do that. As long as you have the software with you, if you can purchase it, you will save a lot of down the line headaches. Then you have the in vitro, which is perhaps the most used in the lab in terms of drug metabolism. We'll go through each of them briefly and some of them in details. Next one is in situ, means the organ has been taken out, but you are perfusing to keep it alive, and you run metabolism so that you kind of mimic or imitate a condition between in vitro and in vivo. And obviously in vivo, we know, is in humans or humanized mouse or primates, for example, dogs, or for example, monkey, and then preclinical animals like rat and mouse, and dog is under preclinical. So under primate, we have things like monkey, gorilla, chimpanzee. Those are the primates. Those are also heavily used in the industry, but in a limited manner because of any issue across the world. But if I say which is largest body of info you get from here so far, this is the area what people do in the lab most often, because in vivo you can't do clinical studies every day with the humans. Humanized mouse, they can be done, but again at the end of the day, they are mouse model which has been modified to reflect human metabolism. And the last 2 in vivo models are difficult in terms of, especially monkey, dog, those are difficult. Rat and mouse are done. But the advantage in the in vitro model, and I do that a lot, is you can have human tissue samples easily and do it to the different cellular fractions. So we're going to spend some time here on this aspect shortly. So in vitro models first. Now, as you know, when we ingest a oral medication, it goes through intestine first, intestinal tract, then it goes through the portal vein to the liver, systemic circulation, and comes back to liver. So it tells you that intestinal tract and the liver both are extremely important in terms of fast pass effect, and eventually they determine the bioavailability of the drug molecule or any chemical molecule. So that's the reason intestine and liver are the 2 organs we heavily study. I will say 75% of the literature in this area is on these 2 organs, liver and intestine, because one is they are the really gatekeeper of drug levels in the body, number one. Number two, most of the cytochrome P4 phase 1 and phase 2 enzymes are expressed in these two organs. That's why it is very widely used in this area of research. Now, in terms of liver, just to give you some perspective how they differ between animals. So on the top, you have different columns: human, rat, dog, and monkey. I said monkey is the primate which is closest to humans, but you don't get the opportunity. Monkey is almost treated like humans when it comes to animal care. So it's hard to use monkey on a regular basis. Unless it's toward the very end, right before humans, that's only you can use it, but not until that time. Here you have the measurements like body weight. You can see this is the average human body weight. Rat, dog, and monkey, they are much smaller. Liver weight, humans has that's in gram. So 1.7 kilogram of liver you can find. That's a huge organ. This is the largest internal organ in humans. And then other animals have a smaller liver. In terms of P450 or CYP content, you see humans are actually lower. Look at monkey levels, almost three times of humans. Rat, dog, all of them have actually higher metabolic content, P450 content, than humans. This is why I think it is important to understand when you do a study other than humans, you need to take care of the species differences, because species differences are extremely important when it comes to what do you call that, extrapolating data. If you have done a study in rat or dog and you want to apply it in human situation, you need to apply appropriate scaling factor. It's called SF or scaling factor, so that you can understand how much metabolism might go on in the humans. So there are a lot of complex calculations behind it, but I want to give an idea that species difference is important. And if you do study in one, you cannot extrapolate just right away to the humans. Now, in terms of CYP enzymes, this is a cellular structure where you can see mitochondria and whatnot. This is essentially the cytoplasm, but the area I want you to highlight are those red ovals: smooth endoplasmic reticulum and rough endoplasmic reticulum. So these are the two main components of protein synthesis, right, or protein house. We know that endoplasmic reticulums are the powerhouse of protein machinery. Out of the two endoplasmic reticulum, the smooth part, smooth endoplasmic reticulum or ACR, are the ones which is responsible for P450 housing. So the smooth ER are responsible for containing P450. That's where they are housed or reside, and that's why we take that particular piece through fractionation, and we prepare different experimental model. Now, we talked about liver and intestine and whatnot. So once you have an organ, you just don't use it straight away, because you want to enrich the P4, the endoplasmic reticulum or smooth endoplasmic reticulum, as you see in these blue color bags, floating bags. So once you have the liver, either from human, or from rat or whatever, you homogenate it. And once you have the using a centrifuge, so then you spin it. Once you have the homogenation done, you spin it and you get something called a S9 fraction, where you run it at 9,000 G in the centrifuge machine. And the supernatant you get, you call it S9 fraction. Whatever pellet you get, you go ahead and run at a much higher speed. There's a very high speed of 104G or 104G or about 100. About you can go closer to that. It doesn't have to be exact, but at least 100,000 G speed. From that, the supernatant, which is essentially on the top, this gray thing on the top you see, is called cytosolic fraction. Now you are getting the cytosol part, and the pellet you got at the bottom here, the red thing in the bottom of the test tube you see, that's called microsomal pellet. That's where you have all these smooth endoplasmic reticulum concentrated. So microsomes are nothing but fragments of smooth endoplasmic reticulum. So I'm going to use this terminology today a lot, microsomes. As you understand, micro means small, zomes means vesicles. So these are the very micro-sized vesicles of endoplasmic reticulum, which is very rich or very high in phase 1 and some of the phase 2 enzyme content. So CYP enzymes, UGT enzymes are very high in those microsomal pellets. And you take it, you dissolve in a sucrose solution and sucrose and glycerol solution for stability, long-term stability. And you put it in minus 80 degrees centigrade. You put it, and whenever you want to work, you take out and you thaw it. Then you dilute it, and then you use in the assay. That's how you prepare the microsomes, be it from human, rats, and so on. Now, these microsomes, the human microsomes, you need to find a way to get it, either from the hospital or from different collaborators. In my research over the last, I'll say about 20 years, I have largely used human samples because that's what you can get closer to human interpretation. But the rat studies are also good if you need to do a particular type of experiment for toxicity and stuff like that. Now, microsome fraction, what you have done through centrifugation, you have reached to, it has advantages and limitations as well, obviously, like any model. It's relatively low cost because if once you've done an animal study, you can prepare a lot of microsomes and apply these for different studies. It's well characterized. We know their behavior, their characteristics really well. Long-term stability. They can be stored in minus 80 up to 25 years, 30 years. My PhD supervisor had showed me samples, and I worked on some of those. Those were from late 70s. Of course, in my lab, I don't have that old because I haven't been doing this independently that long. But I have seen in my PhD supervisor's lab 30-year-old samples, and they are fine. And they work just good. So unless you have some power outage, they are very good. They are very good in stability. Their limitations is you cannot do quantitative estimation because in vivo, there are multiple systems working at the same time. So, and these things are enriched. So that means the reflection you get of metabolism is relatively higher than what you see in human body. Because here you have concentrated. By doing fractionation, by doing centrifugation, you have enriched it. You have made it a very, I'll say, high level of CYP or other UGT enzyme. So it's not a exact reflection of in vivo. That's what it shows in the second point, higher biotransformation rates than humans or even hepatocyte or organ slices, so on. Some of the enzymes are actually absent here. As you realize, in the previous slide, we discarded the cytosolic fraction, we discarded the S9 fraction. So any enzyme, for example, an acetyltransferase and then glutathione transferase and so on, those are cytosolic enzymes. They are expressed in cytoplasm. So You are not going to get those enzymes in these microsome, liver microsomes, because you have left out those supernatant, those fractions. So at the end of the day, you have to compromise some of those phase 2 enzymes. The good thing is these are phase 2 enzymes. So you can still get the full picture of the phase 1 and most part of the phase 2, but some of the enzymes like here, you will be missing. So there's the limitation. Like any model, this also has something in limitations. When you do the reaction with these microsomes, liver microsome or intestinal microsome, you need to give an NADPH regenerating system. So you need to give it needs electron to cause the oxidation and other type of reactions. And that's how you use glucose 6-phosphate, which gets converted to the 6-PG, and eventually you generate NADP plus to NADPH. And that's the required energy source for CYP-mediated metabolism. So you need to provide an NADPH regenerating system. Otherwise, you need to dissolve NADPH powder and then put it and add it through pipettes. So either you give an enzymatic system which will generate NADPH for the reaction, or you add NADPH solution manually. But this regenerating system is better because it can run the reaction longer because it's regenerating, right? Whereas if you add powder or solution-based thing, it is going to run after some time because there is no enzymatic system to regenerate NADPH for a longer period. Okay, but NADPH is a key component of drug metabolism reactions by CYPs and other enzymes. Now the next, remember with the liver, we found 3 major fractions: S9, cytosolic, and microsome. Microsome is the one which is used very often, but S9 and cytosolic also have some role. In the S9 fraction, you get a global view. In the S9 fraction, you have everything. You have the microsomal enzymes, cytosolic enzyme. But the limitation or disadvantage it is a lower enzymatic activity because it is diluted. It is not enriched like the microsomal pellets. So you are going to get a lower enzymatic activity. And that may not be sometimes very helpful in doing your study. That's why we do not prefer usually S9 fraction, because that will make the signal down. Cytosolic fraction has mainly the enzymes which are present in cytoplasm. And remember I was saying earlier, NAD, SALT, and GST, those were missing from microsome. But in cytosol, you have those, but you will be missing the CYP enzymes, UGTs, which are the major players. But still, at the end of the day, these models, S9 and cytosolic fraction models, tools help you to complement each other. You want microsome, you want S9, you want cytosol. That's when you see a full picture. And that's why during drug discovery, people use all the 3 tools in finding out their metabolic profile. Because if you miss one fraction, you might be missing the big picture, or you might be missing the toxic metabolite from the other enzymes happening. So you can't ignore one or the other if you want a comprehensive view. Next one is the intestinal drug metabolism. And I wanted to just highlight that the CYP enzymes, CYP3A4 and so on, they are heavily expressed in the intestinal lumen. So when the parent drug crosses the intestinal lumen, it does undergo a lot of metabolism. So by the time it comes to the liver for fast metabolism, hepatic metabolism, it has actually lost some drugs already by having intestinal metabolism in the small intestine. So that's why we treat intestine as the second most important organ when it comes to drug metabolism. And we use intestinal subcellular fraction, same way as we just talked about liver. And here, the enterocytes or the intestinal cells of the villus are important. You know, all these finger-shaped objects are villus, which eventually represent enterocytes and CYP enzymes there, right? So intestinal subcellular fractions we also use a lot. The next major category, which is not organ-based. So far we are talking about organ, right? Tissue, I mean to say liver, intestine, and so on. Now, in case of recombinant CYP enzymes, I think the name is self-explanatory. It's a synthetic, right? Biochemically synthesized cytochrome P450 enzymes. So it's artificially, you have synthesized by cloning protein using cDNA, complementary DNA. And then you have identical amino acid and substrate recognition ability, just like you see in the tissues or liver and intestine. So the turnover also could be same or different depending on the various expression system. So this is now kind of bit molecular biology. I don't do this part, but I do use recombinant CYP proteins. But I wanted to give you an idea how these recombinant things are developed. So various expression systems in the molecular biology, they are used. Bacteria, yeast, mammalian cells, insect cells, a lot of those are used. But the idea is to use a cell system very different than humans. Because if you use something like humans, there will be impurity, right? So that's why, for example, insect cells are considered very good model for expression. The reason is because insects are very different than us. So they have no P450 enzymes actually, as a matter of fact, or very little, and which are also different than our sequences. So at the end of the day, a simplified CYP expression system will have CYP DNA preparation, transfection of the cell line, multiplication, you allow the cells to grow, you isolate microsomal fraction, you isolate microsomal fraction and characterize the proteins, as we have shown in this particular diagram, that they are like, they start with the cDNA library, becomes mRNA, and then you insert it in the gene you want to synthesize by different enzymes such as ligation and whatnot. So I'm not getting into the molecular biology part here for the sake of time. At the end of the day, this is your kind of, I'll say, modified gene, what you want to synthesize. And you put it in the E. coli, which is a bacterial expression system. And then they grow, they amplify by themselves, reproducing bacteria, right? And then finally, you have the cDNA library and P450 expression. And you do the protein translation happens there, and protein purification, and you get those recombinant proteins into small vials. So at the end of the day, your goal is to achieve this, and that's what I use. I do not use any of the steps before. This is my target, to get recombinant protein. So sometimes I get it from collaborators, sometimes we can buy it from the industry. There are commercial sources of those. But the bottom line is, there's a lot of molecular biology is involved, as you see. But end goal is to synthesize human CYP enzymes along in the E. coli or different expression system, and can use the purified CYPs in drug metabolism assays. In terms of advantages, this one you can prepare isosome specific. For example, in liver, you get all enzymes together. You need to do additional studies to understand which enzyme is doing what drug metabolism. But if you have, for example, CYP3A4 recombinant protein, if you have CYP3A4 recombinant protein and you run a metabolism assay, you know that this particular reaction was carried out by this enzyme. So that's why at the end of the day, they are very straight in giving the answer. At the same time, you can also prepare some genotypes. So pharmacogenomic studies are perfect for these recombinant enzymes because you can produce different variants or alleles, right? Different polymorph forms, and then you can synthesize it deliberately, and then you can find their effect on the drug metabolism. These advantages, you are missing the big picture. It's not like human tissue or organ, and you are dealing with one enzyme at a time. And sometimes there could be structural issue. The way the protein is synthesized, you don't have access to the site of reaction. So drugs cannot, or the molecules cannot approach the enzyme, and you see some aberrant result. Enzyme kinetics data also can be very different than humans. So there are differences, but it also gives a lot of advantages. So you have to evaluate what you exactly want in terms of outcome. This diagram is to show some example how things differ between different enzymes. So this is a whole list of enzymes which I'm not going to speak, but just look at the bottom to see the color coding. The purple ones are the bacteriosomes, like for example in E. coli. Supersomes are the ones which are produced in things like insect cells. And finally, the blue one is the pooled human liver microsome, the ones which have been obtained from humans. Now, just look at CYP3A4, for example. Let's take CYP3A4. This is the CYP3A4 enzymatic activity. And can you see that the blue color is much smaller than the purple? So that means human liver microsomal activities are much lower than the bacteriosome-based recombinant proteins. And the insect cells, it differ between enzyme to enzyme. So my point on this slide is to understand, to appreciate the fact that the recombinant proteins can be slightly higher or much higher than the human liver microsomes, especially the bacterial expression system. So you cannot extrapolate right away. You need to understand the differences between different models. Human liver microsome versus bacteria-based recombinant protein or insect cell-based recombinant protein, right? The next one, I want to give you an idea about the stability. If one of you decide to do this study down the line, you need to know the stability of the proteins, because the problem is if you assume they will be fine over the time, you may get erroneous result. So this diagram, in the x-axis, you have different CYP enzymes, recombinant CYP enzymes, 3A4 to C9 and whatnot. Y-axis, you have enzymatic activity. And different color bars here tells you that how many cycles of freeze-thaw, how many times you have taken out from -80 and you have put it back, you have thawed it, and then you put it back again in -80. And you can see that it's quite actually good in terms of activity. And you don't lose a lot up to 5th cycle. Then also the loss is about 20% or less than that. You can see the last one, the blue color is 5 cycles, right? So you can see it's either 20% or even less some cases, or some cases actually none. So it is a very stable system over a period of time, at least up to 5 cycles. But you need to always keep an eye, because you just don't know how many times people actually thawed it, especially if you have a large lab and different people working with it. This is another example of the long-term stability and different enzymatic activity, same as earlier. Here you can see 98, 2009. So more than 10 years over a period of time. If you take any particular enzyme, for example, let's take CYP3A4 again, because that's the major enzyme. You see how they differ different color. So you can see that over the time they did drop. They did drop. But some enzymes they don't drop at all. So my message here, you need to keep an eye that how your protein samples are behaving over a period of time. We can't assume they will be fine over the period of 20 years, 30 years, because there is a lot of factor. Was there any power outage, electricity shut down or anything? If those things happen, their stability will be less than 12 years, 20 years. But if the power outage was never a problem or nothing like that, they are stable over a long period of time. And this is another data shows that if there is a lot of freeze thawing, the P450 levels can drastically go down. So 0.304 was the level of P450 during freezing, but after freeze thawing multiple times and leaving it on the bench for 6 hours, you see the drop has dropped like anything. It's about tenfold or more actually dropped in terms of protein levels or activity. So far we talked about the recombinant protein, and then we talked about the and then the different tissue-based or organ-based experimental models. Next one is the cell culture. So like any diseases and something, you can also use cell culture in this particular metabolic areas. The most preferred one is primary cell culture or hepatocyte, which is one of the best models, but also very difficult to get because you need to isolate fresh cells from human liver, and you may not get fresh human liver all the time, so it's hard. Secondary cell lines are like usually you see those cancer cell lines, right? HEP-G2 or CACO2, those different cell lines are immortalized. Those are secondary cell lines. Then you have transgenic cell line, means you can modify the cellular genetic structures, and you can have modified version of the enzyme if you need for the experimental purpose. But at the end of the day, you still prepared the same thing, microsome, cytosol, and S9. Like I was saying, human hepatocytes or human cells, liver cells, are considered the gold standard. The reason is, number one, it is live. You can grow them as much as you want, up to 3 times, 3 passages. Then you have both phase 1 and phase 2 in the same cell system. You are not missing anything. If you understand, in the microsome, we were missing some. In the cytosols, we were missing some. But in the hepatocytes, you get all the enzymes you can see in the human body exactly the same way. So that's the best part of it. Now, advantages I said, but the problem is viability or availability. So that means it's difficult to keep it alive. That's a big issue. Contamination of the hepatocytes can be a problematic. Isolation is very complicated and time consuming, and not all labs are equipped for that. And you need good training actually to do that. Using microsomes, you don't need a lot of training. But to isolate hepatocyte, you have to have a good training. And then there can be cell damage during isolation, which can lead to artificially low CYP enzymes. So as much as hepatocyte is a very good system, it has a lot of challenges to make it work in the lab. That's why not a lot of lab works on hepatocytes. There are limitations for that. In terms of cryopreserved, cryo means storing in liquid nitrogen system and so on, right? In a very low temperature, ultra low temperature. So some companies have started selling over the last 10 years saved in liquid nitrogen, and they ship those to you, and you can plate it, or you can use a suspension. You can use those hepatocytes either as a suspension, those hepatocyte or liver cells, or you can plate them in the cell culture plate. At the end of the day, it's more convenient, but also they are not fresh cells. They are in between secondary cell line, like cancerous cell line, and the fresh liver hepatocytes. So there's a compromise between two extremes, but still it's getting very popular. But some of the levels and everything can be modified because it has gone through some storage, right, in minus 180 degree and whatnot. So it has gone through some stress. So you will not have the exactly same as fresh cells. Secondary cell lines, I think I said they are easier. Secondary cell lines are typically cancerous. So that's a problem also sometimes. They are not the normal human cells you are looking at. So because they are cancerous, they're easier to grow. As you know, cancer cells grow well, right? But at the same time, they give some in vitro human data. So people sometimes, if they like it, because it's easier, convenient. And if someone knows cell culture, cancer cell line growth or other cell line growth, they can grow these cell lines. But the problem is they are cancerous, so they are not normal cell lines. Secondly, the enzyme levels are very low sometimes. The enzymes and transporter levels are very low sometimes. And often you will not get the metabolic profile, all the metabolism. I personally worked with cell lines at the beginning. I did not like it. And after that, I tried to stay with either human models, human tissues, or hepatocytes, and so on. But I tried to be away from secondary cell lines because the results are not very good. If you want to do some molecular biology work, then it's fine. But my work is more in the drug metabolism and PK, more in the activity part. So my work doesn't work well with the secondary cell lines. In situ models, the models which are essentially you isolate a liver and you try to keep the functionality of the organ alive by perfusing, by circulating some kind of specialized buffer system, like Krebs Henslett buffer. And by that process, then you run through the drug, through the perfusion, and you get metabolites. So it is kind of, you are mimicking a human situation, because you have human or rat, but typically this is done with the human models or human tissue, because, for example, someone has died, a patient has passed away, and the family is ready to donate the organ. So you take the organ and you keep it alive. You keep the liver alive for a few hours. It's a very short period it will be alive. But you need to keep perfusing with the buffer and other electrolytes and oxygen and everything, so that it will behave like a human liver system in the body. So, but that's also very delicate, and viability period is small, very poor reproducibility, because it is going to differ from person to person, lab to lab. I personally haven't done this, but I have seen others doing that in front of me. But it's a very rarely used, to be honest, because of some of the challenges, and availability of organ is not easy. Nobody wants to waste a good liver for research purpose, rather than they will give it to some liver transplant patient, right? Organ transplantation is much more priority than doing research. Then also it's a very, sometimes it's a very urgent situation. So that means you need to be ready with your work. So this is, I will say, one of the least used models. And finally, in vivo models, means which are inside the body. Not a lot of models we use in this area, except humanized mouse. So you can change the mouse CYP genes and add human CYP genes. So that when you give a drug, that mouse metabolism, once you inject or give orally a medication to the mouse, the metabolites you see, they will behave or mimic human situation. Similarly, we have also knockout mouse models, because if you knock out a particular CYP and you try to identify its role in an enzymatic reaction, that's a good way to be confirmatory in an in vivo system. This is a very good and very commonly used. If you have availability, when availability is an issue, and they can be expensive because they are genetically modified, they need special attention. So, but humanized mouse means you have modified a mouse to mimic the human seeds, can be very useful and helpful in making things functional in a physiological situation, not in the in vitro situation. Now, in terms of steps of a drug metabolism assay, for example, I have taken an example. This is a real study we did about 10 years back. We have some paper from that. So you want to treat a drug with dexamethasone. You have a mice, CD1 mice. For example, you want to see the effect of short-term dexamethasone. So you give that for 3 days, 68 to 62 days, and then you euthanize and isolate the liver. Now, once you have done the isolation of liver, you have prepared microsomes. You have went ahead and, for example, you incubate with NADPH some molecules such as vitamin D. That's what one of my research areas, and some phosphate buffer. Subsequently, you stop the reaction with a cold organic solvent such as acetonitrile, and then you add an internal standard for analytical purpose. You extract the organic layer in the vacuum dryer in the centrifuge, and then you dry it in the vacuum dryer, and then you reconstitute in methanol so that you can inject it into the analytical system. I work on LCMS, liquid chromatography mass spectrometry, because mass spectrometry is a very good system for this, but HPLC can also be used for this purpose. And once you inject that in the HPLC or the LCMS system, you identify metabolites and compare it with authentic standards, and then you do whatever is your purpose, drug interaction study, toxicity study, whatever is your purpose, you go ahead with that. But the bottom line is, once you have done isolated the microsomes from human or animals, you go through these steps, and your endpoint is usually a metabolic reaction by using some analytical tool. You need an analytical tool to do the drug metabolism assay, either LCMS or HPLC or GCMS. GCMS is not that popular anymore, so people use either LCMS or HPLC. There are always variability in drug metabolism studies because here is humans or animals involved. So inter-individual variability you have to always be careful of, and always keep an eye on that. There could be batch-to-batch variation of microsomes. There could be vendor-to-vendor variation, commercial sources. So you need to have a standardization model. For example, each time you get a batch, you need to run some assays to understand what are the benchmark or the characteristics of that batch. Otherwise, you will see after 3 years, 5 years, you have trouble. You can't reproduce the same data what you did 5 years back, because it's not your fault or the student or the fellow who did. The problem is this batch is different because all humans are different. And everybody is exposed to different things like diet, smoking, and other lifestyle issues. So you need to always keep a quality control in the lab when you have new batches coming, so that you know later on the problem or the limitation or comparisons you can do. So I think I have come to the stage where we will be talking about the in silico part. But before we get there, I wanted to see if there is any question at this time, and then I'm going to walk you through the in silico part of it. Like some of the drug metabolism, PK and DDI, you can do using actually computer software. And this is a very upcoming area which started about, I'll say about 10 years back or later. Any questions at this time for me?
Participants, you can write your questions if you have any question. I have a question, Dr. Subrata Dev.
Sure. Yeah, please go ahead.
Yes. So many drugs are having a low bioavailability, and one of the main reasons is this first pass metabolism.
Yeah.
So there are approaches by which we try to enhance the bioavailability, but I don't think there is any control over this first pass metabolism. So what are the various delivery approaches by which we can improve the bioavailability for drugs showing extensive first pass metabolism?
Right, right. No, that's a very, I think, interesting question in terms of the practical part of it. Typically, okay, so in drug metabolism, I haven't gone through the chemistry part. There is also called something soft targets. So there are some functional groups which are easily targeted by these CYP enzymes. So, by using prodrug or conjugation approach, if we modify those, then the CYP enzymes are unable to break down those linkages. But at the same time, once they reach to the systemic circulation, there has to be other enzymes which can break down those conjugates, right, or prodrugs, so that free drug is available for functionality. So typically, if we want to take a uh, approach, chemical approach, I think prodrug or conjugates are very popular. But in terms of drug delivery, for example, polymeric encapsulation and on other stuff, I think they will have a similar issue unless you have some enzyme protective groups present there, or linker present there, some polymeric linkers. Otherwise, people have often tried to change the functional groups, or make prodrugs or conjugates with polymer to protect them until they reach to systemic circulation.
Yeah, thank you, thank you. So I think you can go ahead with your software part. Then at the end we can take some questions from the
Sure.
participants.
Sure.
Sir, one question is there.
Yes, yes, please.
Some delegates are doing this program through YouTube. So in YouTube, one Chandrakala asked the question like this: Sir, is our kidney may damage when we use long-term therapy due to filtration by kidney?
Typically not, unless there are some toxic compounds. Now, this is also one aspect of, after liver, intestine, kidney and brain are the next two major sites of drug metabolism. So unless some toxic metabolites are forming, because kidneys are meant for filtration of huge amount of water daily, liquid form. Now, unless there are some toxicity or cellular damage from drug metabolites or the drug itself, there is no reason over filtration is going to cause damage. Now, if someone is drinking, for example, just for an example, 50 liters of water a day, yeah, obviously that now the whole body is under stress. And someone actually can die from drinking water if someone drinks a very, very large amount, like 50 liter, 100 liter of water in a day. But typically, a regular amount of water, or even 3 times, 5 times more water than what we drink, it's not going to cause any issue with the filtration system, unless the drug itself is toxic or the metabolites.
Thank you, sir.
Thank you. Okay, so I'll go ahead, and again, I'll be available at the end. Yeah, go ahead.
Good evening,
Yes,
sir.
please put your question, and then we'll proceed for the software part.
Yes, sir. Good evening, sir.
Sure, go ahead. Good evening.
Sir, I wanted to ask a question regarding, so there are some drugs like atracurium which undergo Hoffman elimination reaction. That is, they undergo metabolism without any elimination, means without the role of any metabolic enzymes.
Mhm.
Sir, how does that Hoffman elimination take place? Means how there is no role of any enzyme for the metabolism of such drugs?
So what was the name of the drug you said?
Sir, atracurium.
Okay. So there are some of the reaction are spontaneous reactions. So I don't know about this particular reactions, whether it's happening, but typically when we say there is no enzyme involved, we often say that means no traditional drug metabolizing enzymes are responsible. Otherwise, uh, in 99.9% cases, as far as I know, there are some enzymes available. For example, esterase or hydrolases, and there is a whole long list of, for example, aldehyde oxidoreductases. So a long list of enzymes are present which can catalyze, but they are not studied and referred and often said as spontaneous reaction or non-enzymatic reactions. But this particular example, I'm not sure how it happens. Have you done some literature search in terms of if there's any enzyme available or any non-traditional or non-metabolizing enzymes also available for elimination of the drug?
I think you can go ahead with your presentation. At the end, we'll see.
Okay. Sure. So for now, I'll go ahead for the interest of time, and I'll be always available towards the end to speak more about any other questions or follow-ups. So you can see my slides again, right?
Yes, yes.
Okay, thank you. So this in silico or computer-based software can be used for several purposes, but I'm just showing today what I use for. Um, Drug metabolism, pharmacokinetics, and DDI. This is the three areas I work. That's my key research areas, both in the in silico part as well as in the lab part. And there are at least two softwares came up in last about 8 years or 10 years. And I have listed these two names here. One is GastroPlus from a company called Simulations Plus, and the other one is SimSip from Sartera. And I use GastroPlus, that's why I bolded it, and that's what I'll be showing in next a few slides, and then we have the software to show. But I wanted to show that if SimSip is also an option, but GastroPlus is the one which I use. Now, if you go to the website, you can do it later, but I wanted to show that Simulations Plus, if you go, there are several resources present in this particular area, and they are very good in putting training stuff and self-learning material in this aspect. And I must tell you, I did not get any training on that. My PhD was on purely lab-based thesis and work even later. So even my postdoctoral training was on drug metabolism and human research. But this one I started working about 4 years back, 3.5 years back, 4 years back, when I came across these software developers in a conference, in AAPS conference, and that's where I eventually submitted a proposal, and they funded my research, and they give me actually software access. Licenses to carry out my research. Obviously, I have to give yearly progress report and everything. So this is the software website, Simulations Plus, and if you click on the software, then you have to choose Gastro Plus from that list, because they have other ADME software. They have a bunch of ADME software, means absorption, distribution, metabolism, and excretion software. But GastroPlus is the one which I work on. Now, if you go into the GastroPlus part, these are different modules of GastroPlus, which is used. And like I said, I use drug interaction, that I call it DDI. I use ADMET predictor. As you know, ADMET means ADME plus toxicology. So ADMET, it can interpret some of the things we'll talk in a minute. And then I use PBPK plus, physiology-based pharmacokinetics model. That's what I use for these three things. But if you look at, I also actually use metabolism and transporter to some extent, but that one is already covered in the other three modules. But you also have other areas. For example, I know a lot of people from pharmaceutics are very benefited from things like IVC, in vitro in vivo correlation, and so on. So they have a wide range of software which is used. From time to time, they do actually free conferences. As a matter of fact, in couple of days, there is a conference coming up. If you go to their website, there is a potential, they have space. I am attending this actually virtual conference, but this is what they do in terms of GastroPlus. Now, when you log into the software first time, you come across this window. You can see different tabs here on the top: compound, then gut physiology. I'm going to show them in a minute, all those. And then you have the pharmacokinetics, simulation, graph. So as you change tabs, you can see different types of activities there. But the first part is, once you log in, this is the part where you import the structure, and through that process, and you are activating the module called ADMET predictor. Once you import the structure, and can you see here in the bottom, the properties and predictions are for ADMET predictor. Once you import the structure, it's going to predict. For example, one of the medicinal chemists tell me, oh, I have designed a molecule. I am unsure. Should I go ahead and synthesize it? I can tell, looking at the analysis, oh, you may get a low bioavailability. You may have a problem with the solubility from all these things you see in the middle, where the pH and all the different log P, all these physicochemical properties can be interpreted. Then you can have perfusion coefficient. All these, whether it will have a good permeability, means bioavailability and solubility, everything. So some of the biopharmaceutics characteristics can be interpreted by using this particular module. It's called ADMET predictor. Okay, you can also determine the dosage form, which dosage form you want to use, in immediate release you're going to use, and so on. Then you go through the PK and other modules, and eventually you come to DDI part, where you have one component for victim drug, means the substrate which is getting affected, and the other one you get perpetrator, means the drug which is causing problem. So these are usually inhibitors or CYP inhibitors or inducers, and these are usually CYP substrates. And you are trying to understand if the inhibitors are going to impact, if the metabolism can be blocked or not. And eventually you see an outcome. Here you can see in the steady state control of the drug levels that the perpetrator can strongly actually inhibit the CYP metabolism of a particular drug. In this case, we are talking about calcitriol, means it's a vitamin D derivative. And that way, if you give this drug A and drug B, there is a good potential of interaction even before you go to the lab. And that is helpful because you can design your experiment accordingly. As you know, in silico method is never foolproof. You need to validate the results by experimental data, but it gives you confidence and time-saving and cost-saving methods to find out when you work with the new molecules or drug-drug interaction therapeutic scenario, can there be any issues? For example, in a lot of population, you cannot have clinical trials, or it's very hard to do. Pregnancy is one situation. Pediatric patient, another situation. So what you can do here in this software, you can change the physiology of the virtual patient, hypothetical patient, based on the pregnancy and pediatric situation. And you can run this pharmacokinetics and DDI study and try to understand whether there will be interaction in the individuals. Because as you know, as our body physiology changes, the ADME changes. If the body fluid level is high in pregnancy, for example, the volume of distribution will change. So that means it's going to change the PK parameters. So it is going to change the DDI at the end of the day, drug-drug interaction, bioavailability, all these aspects. So through this model, you can modify it according to the physiology of the patient group or the target population you are trying to give the drug. So that's what it helps you to give you the an insight even before you start the real experimental work, or you want to give it a snapshot before you try the drugs on population. So now I'm going to share the actually screen for. Okay. Can you see the? Can you see this window now where the software is there?
Yes, yes, we can see.
Okay. Okay. So here I want to show you how you can import. So this is the same page where we same place where we were at the beginning. So you can import the drug from this database. So that's what you can see through the. If you look at the cursor here, we are trying to pick up the drug from the database we create. You have to create that database. So we pick here simvastatin, for example. And as soon as we pick simvastatin, this is a known drug, so that's why I'm showing as an example. Different properties can be found here. So you can select whatever you want, ritonavir, whatever drug you want, carbamazepine, and in the bottom you can see all the different characteristics of the bioavailability and whatnot characteristics. But you also see here all the solubility, all the effective parameters. This is real data now. You are seeing log P, and that's how you get the ADMET predictor outcome through this software. Once you have done this step, you go to the next, and then you can also change. Can you see all these different options? Dispersed system, topical solution, all different kinds of powder, infusion solution. You can change the type of dosage form. And based on that, the ADME properties will change. Physicochemical properties are not going to change, but the ADME properties are going to change. And you can also check here. You can see right-hand side of the page. You can change the species: human, rats, and so on. So this platform gives you different options to work with dosage forms, different species, different drugs, so that if you have a new molecule, and then you know what kind of expectation you can have in terms of ADMET predicted before you go to the lab. And once you have done and understood the predictions from the ADMET part, you go to the next step, which is the gut physiology. And you can see here in this particular aspect, the gut physiology part. I'm just trying to enhance a bit here. It tells you at the bottom, you can see, it tells you the metabolism by different enzymes, CYP3A4, and the transporters you can see listed at the bottom here, how they are will be playing a role in its ADME profile. Okay, so there's the ADMET predictor part. Now, next step, gut physiology here. I want to show you the gut physiology one second. So here you can see that you can change the gut physiology. You can change different parameters of the pH, transition time, all the biopharmaceutics, whatever we learned in our career, you can change all these options, and so that physiology of the patient you are mimicking the way you want. If you have someone obese, if you have someone liver dysfunction, renal dysfunction, you can change all of those here. Okay. Um, and then the next one is pharmacokinetics domain. Here, now you see the tab is highlighted pharmacokinetics. Here you can find out all different kinds of PK parameters: bioavailability, clearance, volume of distribution, AUC, Cmax. All these components are interpreted here, and how you get it. So you have to save it, and then you can change the physiology of the patient again and decide you want to human, dog, rat, how much weight you want. So all this body weight, body fat. If you look at the left-hand corner, bottom corner, you can see height, weight. You can change the patient's particular. So you can actually customize the way you want, because that's what you want, individualized data for each patient, especially when you are in the hypothetical or in the in silico system. You can also change population. You can put American, Asian, gender. So you have a lot of options to change to reflect the population you are trying to work with. Then next, you have the simulation part. That's what we will be going to. You have to save the patient here because you have created a new patient for yourself, and then you will be going in this part. So here, if you look at, I am clicking on the start, and it is calculating now. If you look at the numbers are changing because whatever structure you gave, whatever gut physiology you have given the condition to the for the patient, and now it is running the simulation, and it's telling you all these different PK parameters by calculating, computing, and going through different parts of the GI tract. Okay, so that's the PK part of it. And the last one is the DDI component. And this one, I think I gave you some idea. Again, here you see simvastatin in the left-hand side as the substrate. And in the right-hand side, you have diltiazem, which is the inhibitor. And you can change it based on your database, whatever you want to use for. And then you run a different simulation by, of course, you have to go through some steps, understand the requirements of the software and the protocols of the software, and at the end of the day, you have to choose the right kind of combination. For example, if you have data from lab experiments like human tissues, you use as in vitro data and run it, and eventually you see an outcome. For example, like this. If you see here, it's saying strong or moderate, systemic Cmax or liver. So you can either understand the interaction in the systemic circulation, in the bioavailability, or if the interaction is happening in the liver, how it will be. So, and the classification can be either strong or moderate, and that's how it will give you some idea. If you give simvastatin along with ritonavir, ritonavir is a very strong CYP3A4 inhibitor, and simvastatin is a CYP3A4 substrate. That's why you see a strong inhibitor here, strong inhibition prediction. And once you go to the lab, if it's an unknown molecule, you want to experiment that. But in this case, we know because I took them as an example. So simvastatin and ritonavir both are known drugs. But if it is an unknown drug, it gives you the idea even if before you go to the lab, right? So that's the beauty of this, eventually the model. So So I think that's essentially, and then you have to select what type of simulated concentration you want: liver, gut, systemic, all of those. You want the concentration of the blood, liver, or intestine. You need to select from here, and eventually that's going to help you in getting the right combination and make sure you run the simulation with the right type of matching, and you get the out. You can see there is a calculation going on. So essentially, this is exactly what I wanted to show in terms of uh, the capabilities of the software, and of course, the purpose of this walkthrough is not to make you an expert, as you realize, but to give you a flavor what can be done. If you are interested to do more work in this area, obviously, I can be of more help. But let me go back to my other slide to finish off. Uh, so, so essentially, I wanted to show that last piece. I know I have gone bit over time, and my apologies for that. I think it's slightly over time than what I was supposed to have. So with that, I wanted to thank everyone for this opportunity and patient listening. At the end of the day, I know it's evening there, and I am more than happy to take any questions or comments. I'll be staying here as long as you would like to know more. Thank you.
Sir, good evening, sir.
Good evening again.
Sir, that GastroPlus software, if I have prepared some dosage form, taking some active ingredient API, and I have prepared some dosage form, and after that, what means in what manner I can use this GastroPlus software? If I have prepared the, you are telling that means in that software you are showing the API, but if I have prepared the dosage form, I have converted to some liposome or nanoparticle. Is there any use of this software to calculate
I think, yeah, I think it can compare between dosage forms, but I think the specificity may not be that much if you go to liposome or nanotechnology or nanosomes or something like that. Because, for example, you saw in the drop down there are options like immediate release. It will give you a general idea what is the difference of this molecule between immediate release and control release, between topical form, parenteral form. But it may not give the specific information you will be looking for after you have developed the dosage forms, right? So that's the part I'll say. This is more for early stage work, drug discovery and development, more early stage, to decide, do you want to inject the animals or the models by oral route or intravenous route, for example, right? But again, it is helpful later during the drug-drug interaction part in the DDI module. But for the specific question you said, I feel it may not give the exact info you are looking for to understand what is the bioavailability of liposomes or nanosomes compared to a traditional molecule. It will give a general overview, but other modules such as IV, IVC, in vitro, in vivo correlation, and those kind of modules, which I don't use a lot, those might be able to give you additional info. But I'm pretty sure there is something in that area. It's just that the modules I use is perhaps not a good one for that purpose.
Again, sir, another question. We are actually using rabbit model in order to calculate this viability parameter, and we are comparing with the API. And pharmacokinetic parameters which we are calculating the AUCs, Emax, we have these parameters we are calculating, and we are comparing with the API. whether we can compare for a sustained release dosage form and a API. Means whether a formulation we have prepared in our lab, it is a sustained release formulation. Whether we can compare the AUC, particularly my question is regarding bioavailability. Whether we can compare bioavailability of a API and a sustained release formulation, which we have prepared in our lab.
Yeah, that's for sure. That's for sure, because and that's the purpose to select the doses from early on during the preclinical stage, and that can be done because most of the time we run in the immediate IR form, immediate release form, but other forms can be run and compare. Okay, what is the difference between solution form versus tablet form versus control release form? So yes, that difference in PK is absolutely doable with the PK Plus software, which I use.
One more last question, sir.
Yeah, sure, go ahead.
We are finding out two parameters, AUC T to 24 hours. If my objective is to deliver the patient once a day, AUC T 0 to 24, so 24 hours. Another parameter we are finding out 0 to T to infinite time. So what it means actually, AUC T to infinite, what it means actually. In some lecture I have listened that that indicates plasma zero concentration of drug in plasma. Is it the right way? Means is it the
I think it's kind of your interpretation is kind of in the right direction. So 0 to 24, we always want to give a boundary to compare between molecule to molecule, right? Otherwise, or dosage form to dosage form. But some molecules, they need, for example, 5 half-lives, 3 half-lives to get rid of the body, right? And the AUC, my measurements might be done until 48, 72 hours. But often, a lot of studies don't do that. After 24 hours, they just come after, maybe straight after 72. So at that point, you say infinity because you just don't know when that really went down, right? So essentially, infinity means when it is going towards zero at the end of the day, after the dosing. So that's how. So your interpretation is correct in that sense. That's why I trust more 0 to 24 because that is more quantitative, structural. But 0 to infinity is a lot of measurement variation from lab to lab, protocol to protocol, right? So, but it gives some idea how often the molecule can be, or how high or low can go eventually within a certain time period, right? So,
thank you, sir.
Well,
there is a question from Dr. Bimla Tripathi. Madam, please go ahead.
Hello, sir. Good evening, sir.
Good evening, madam.
Sir, my question is, what is the cost of GastroPlus software, sir?
Okay, so I essentially get funded by the from the company, but I know in academia it's about one license is $10,000 in US. Okay, so, but don't worry about it. I'm only telling the US number. I'll come to the Indian in a minute. So about 3 years back, it was introduced in India. There was a conference in Bangalore. That's why I was also part of that conference. Initially, it was Electrolab, which was supposed to distribute, and I think there is a speaker from Electrolab, right?
Yes, right.
Yes.
But I am not sure. I'm not sure if Electrolab is the distributing company in India right now. But in short, if I to give the answer in Indian currency, I don't know what is the price right now. But let me also tell you, if you have a solid research project proposal, they have the ability for academic setup to fund it. They want their profit from the industry, not from academic people that much. But they want the software to be used for good research so that they can utilize that for their own, development, because they have a lot of funding from FDA. If you go to their website, you will see. I try to follow them on LinkedIn. You will see their activities there. They're growing heavily. So if someone is interested in doing it, other than collaboration, the option will be to develop a solid project and then try to reach out to Indian distributing partners. If not, because the VP of the actual company in San Francisco, that's whom I know from the beginning. So that's John DiBella. He's the one who is essentially looking after the whole thing in India. So if you have interest in accessing or doing or whatever, I think I can link you with the right people, and eventually from there, depending on what you do, potentially it could be there could be an option of funding from GastroPlus through the license.
Thank you, sir. Thank you so much, sir.
Oh, welcome.
Good evening, sir.
Good evening.
My question is any online training program is there to learn how to run the software?
Oh yeah. I think today there was not enough time, but if you go to the resources tab, if you go to the resources tab, okay, let me go to the software so that I can at least show something, even if it is not. Sorry. Here you can see the resources, right? Resource center. So if you go here, sorry. If you go here, there are a lot of videos, a lot of articles, and again, software is something like that. It's hard to teach anyone, right? Someone can give you the link. I mean, link with that resources. If you spend time, you know more. But I will also acknowledge my challenges because of time. Since I was not trained during my PhD, I had to learn it, and it took me a bit longer than in PhD. What I could learn in 1 year, it took me probably 4 years, because you can't give full-time time to learn something new, right, at this stage. But if you have time, and if you are not an administrator, you should have more time, because administrators, especially in this part of the world, has just too many different things. So you should have time, and I believe it will help you, these resources. And you can also download a demo version from the website, but it will not allow you to go very far. It will not allow you more than ADMET predicted. But at least the initial part, it will give you the flavor in the demo. And then once you have the license, then you can go to the PK module, DDI module, and all the other modules. But there are a lot of resources there on the website. And also there is a conference coming up actually day after tomorrow. So if you go to the website, you may be able to still register, and hopefully there are some slots available, at least to get some idea if you like it or not, right, before you go further into it.
Sir, another question. Sir, is any free software is available, alternative to GastroPlus?
There are, but those are, I'll say, not user-friendly. Or, for example, MATLAB. MATLAB is a relatively older, maybe about 15, 20-year-old. But there, sometimes it can be complicated and so on. But if we are talking about simulation of ADME, PK, DDI, very limited option. Drug designing, we all know there is a lot of chemical softwares out there, free softwares, right? But simulation, this area is so new. Anybody who will develop, they will not really put it on the market as a freeware right now. But MATLAB is something closest, but it is not user-friendly. I tried a few years back and had a look, but I didn't use. It's not very user-friendly. But I'll say the best bet will be either. And by the way, if you think about SimSip, the other software I said, SimSip is more expensive, and SimSip are not very academic-friendly. They don't fund that many academic projects compared to GastroPlus. GastroPlus has interest in supporting academia as funding. So SimSip is another option, but they are more expensive. So really, the go option is, once you find interest, to find a way to get support from them, at least initially for some year, at least a year or so, and then and then see whether you like it and you can produce something. They may fund it in the longer run, or you may find it through AICT, or I know in India right now, a lot of opportunities in terms of funding, small project funding. They may be able to do that. It's not very expensive, I believe, for academia, especially in India, because it will not be an equivalent of dollar amount. It will be much less than that, much less than that. What is price is here.
Okay, thank you, Dr. Subrata Dev, for giving a very good concept about metabolism and also giving a flavor of this GastroPlus simulation software. Thank you very much for giving your valuable time and sharing your knowledge with all the participants. There is a small token of appreciation in the form of a e-certificate. Please receive it on behalf of Roland Institute of Pharmaceutical Sciences. Kanu sir, can you please share that?
Yes.
Shall I share?
It's 2022.
Sorry, is it visible?
Yes, I have already shared. Okay. Thank you, Dr. Subrata Dev, for giving your valuable time. Really, it was nice to interact you after a long time. So, good night to all the participants, but good day to Dr. Subrata Dev, because he has to join to his school and he has to continue his work. Thank you very much.
Thank you very much, and I wish you all the best with that training. I know today is the first day, and all the best. And yeah, so feel free to reach out if you have any additional questions. Thank you.
Thank you. Thank you.
Bye.
Very much.
Bye.
Thank you for the informative session. Thank you.
Thank you. All the best.
Thank you. Thank you all participants for your patient hearing. So tomorrow again we'll meet at 11 o'clock in the morning. Please join for the next session.